right, so in chapter 3.3, we are now going to talk about prokaryotic cells. So we're going to talk about the unique characteristics of prokaryotic cells. So in other courses that you should have taken prior to this course, you probably have gone into much detail on the characteristics of a eukaryotic cell. So we're not going to go over the specific characteristics of a eukaryotic cell in this course. What we're going to focus on are the prokaryotic cells. So do make sure, however, that you brush up on your information regarding the eukaryotic cells so that as we move through the semester and we're comparing eukaryotic to prokaryotic, that you are familiar with and kind of have refreshed your memory on the eukaryotic cells. So between the two, however, with all cells, there are different things that all cells contain. And the things that all cells contain are, one, they contain cytoplasm. So remember that cytoplasm is that kind of gel-like goopy stuff that's within the plasma membrane. So our gel-like substance of water, it's made up of water and dissolved chemicals. And this is needed for growth because within our cytoplasm we have a lot of our chemical reactions taking place. We'll talk a lot more detail about that, but this is where we have these chemical reactions taking place. We're shuttling things back and forth and around, and including moving the cells themselves. Some different mechanisms are here in the cytoplasm. So the cytoplasm is important in both eukaryotic and prokaryotic cells. The second thing that all cells contain are chromosomes. So notice here that we say chromosomes, and that's because these are the genetic blueprints of the cell, and they are different between eukaryotic cells and prokaryotic cells, and we'll get into that in just a moment. But so remember that chromosomes are going to contain the genetic blueprints. Genetic blueprints of the cell. So all of the genes are contained within the chromosomes. And of course, in eukaryotic cells, we have many chromosomes. In prokaryotic cells, there's a single chromosome. We'll talk about that. The third thing that all cells have are ribosomes. So you should recall from eukaryotic cells also, your study of eukaryotic cells, that ribosomes are the organelles that make proteins. So organelles used for production of proteins. So you should be familiar with these different things, the cytoplasm, chromosomes, and ribosomes. So already you know those three things. Moving into our prokaryotic cells as we move into more detail. So all cells contain these when we're talking about throughout the semester, eukaryotic or prokaryotic. But we are going to talk about some differences. So recall that if we say prokaryotic, what we are meaning is that they lack a membrane-bound nucleus. So they lack a membrane-bound nucleus, and what they have is a single chromosome that's located in what's called the nucleoid region. So they have a single chromosome that is circular, located in a nucleoid region. So again, since they all have chromosomes, I spelled that wrong here, let me fix that. So nucleoid, let's get that correct for everybody here. Nucleoid region. Nucleoid. All right, so our prokaryotic cells, again, they lack a membrane-bound nucleus, but of course they do have chromosomes, so they have a single chromosome that happens to be a circular chromosome, and that's located in what's called the nucleoid region. So again, it doesn't have anything containing it, keeping it within something like the plasma membrane. So it can float around the cell, but it all kind of sticks around itself, is curled and twisted, similar to our eukaryotic cell chromosomes, and they stay in a region called the nucleoid region. So then conversely, if we talk about our eukaryotic cells, again, what we're talking about is that they have a nucleus that is surrounded by a complex membrane. So that's our plasma membrane is surrounding our nucleus. And then in this case, the nucleus is going to contain multiple rod-shaped chromosomes. So rather than having one single circular chromosome, in our eukaryotic cells, we know that we have multiple, contains multiple rod-shaped chromosomes. So if we recall what that looks like, we can kind of do our general drawing something like this, where we have the attachment here for our sister chromatids. We often see them like that when they're duplicated, but when they're just their unduplicated state, they look something like this, and we have multiples of those in our eukaryotic cells. Our prokaryotic cells, then we have one single circular chromosome, but it doesn't just look like a big circle, it's all kind of twisted around and wound up on itself. And it's still a circle, but it's all kind of twisted up there and condensed, just like these are condensed. And if we look at some differences in our prokaryotic cells compared to our eukaryotic cells, what we can see is that our eukaryotic cells tend to be larger than our prokaryotic cells. So these are smaller. And again, we have our membrane-bound organelles. In our eukaryotic cells, so not just the nucleus is membrane-bound, but we have other membrane-bound organelles in our eukaryotic cells. Whereas over here, we lack our membrane-bound organelles. Also in our eukaryotic, remember that when we're talking about domains, what domain are we in here? Right, domain eukarya. What about prokaryotic cells? What domain are we in here? Got two domains in our domains, archaea and bacteria. Good. All right, so let's take a closer look at our prokaryotic cell. All right, so here is an image from your text, from the OpenStax text of microbiology, and you should be familiar with this image. You should be able to label all of these different structures and define the structures or give descriptions of the structures. Right now, I just want to kind of talk through each one of these things, and then we're going to go into much more detail over the course of this section of the chapter here, after we talk about some morphology and arrangements of cells. So if we take a look here, if we look kind of on the outside of the cell altogether, start from the outside and work our way in, we can see that we have the capsule here. And we have already spoken about staining techniques, and recall that we did talk about staining techniques regarding capsules. And so this is why we would see that little halo around the bacteria, because we would be staining the inside there, staining the cell wall, which is our next layer that is deep to the capsule. So we would be staining our cell wall, and then we would be staining the background, which would be all along the outside here. And so what we are seeing with that halo is actually this capsule that you can see that's relatively thick if you look at the illustration here. So relatively thick, especially in comparison to the cell wall or the plasma membrane. So the capsule, again, is going to help to make things more virulent. It helps to make them stick to things, grow in groups, and other things like that. That we'll get into more details. Then deep to the capsule, we have our cell wall. You can see the thickness of the cell wall comparatively in the illustration. So we have our cell wall. We're going to talk even more about the cell wall and the peptidoglycan that we've already discussed, the different layers of peptidoglycan, how much or how little, and some other aspects of the cell wall that are important to identifying are gram-negative and gram-positive, and also some of the linkages between them. And then also remember when we talk about archaea, we have our pseudopeptidoglycan. So we're going to look at that in more detail as well. Deep to the cell wall, we have our plasma membrane, even thinner. So the plasma membrane is just like we know the eukaryotic cell plasma membrane to be. So made up mostly of our phospholipids, has the cholesterol embedded, the proteins embedded to allow for transport between and throughout the cell. So very, very similar in structure to the eukaryotic plasma membrane. Then if we move deep to that, then we can get to our cytoplasm. So again, that gel-like substance that we find in cells where all of our chemical reactions take place and basically where everything is within the inside of the cell, kind of floating around in this gel. So some of those things that are floating around in the gel would be ribosomes, for example. Another thing that we find in all cells that are going to be the organelles that are going to help us to make our proteins. And this is similar to what we've already studied in eukaryotic organisms, where we undergo the process of making proteins from amino acids. We already briefly mentioned the nucleoid region. So there is that single chromosome, circular chromosome, that's all kind of wrapped around itself in that nucleoid region. And then we have inclusions, which are kind of like other vesicles that maybe you've discussed with eukaryotic cells. So some vesicles that you may be familiar with are lysosomes or peroxisomes. So inclusions are similar in that they are vesicles, but typically inclusions are filled with other things that we're going to get into much more detail. So sometimes they're magnetosomes, which have magnetic capabilities to allow things to move in regards to different poles. Sometimes they're filled with things that the cell may metabolize. So we're going to get into that in more details on the different types of inclusions you need to be familiar with. Then we have our plasmid here. Our plasmid is extra chromosomal DNA. So we'll talk a lot about plasmids in this chapter and throughout the semester. So it is separate from the nucleoid region. It doesn't contain any of the, or generally, it doesn't contain any of the major genetic material or major genes that are required for that cell to continue living. But it confers other qualities to the cell itself. So sometimes that's where we would see, in particular, our antibiotic resistance genes, for example. So plasmids are very important. And this image just shows a single plasmid, but bacteria could have 0 plasmids, 1, 2, or up to hundreds of plasmids. So it really depends on the bacterium. If we move back to the outside of the cell, then we notice that we have 3 different types of cell extensions here. Up in the top, we have our fimbria. And our fimbria, as you can see, are kind of all over the cell. They're like little hairs. They also work similarly to the capsule, where they allow things to get kind of sticky. I think of them kind of as little hairs all around, but less than hairs. I think of them as being kind of like Velcro, little tiny, tiny extensions that help to stick to stuff. We also have a pilus, which is singular that they're showing here, or pili, which would be plural, several pili or pili. And those also are going to assist with attachment. And we're going to talk specifically about the F pilus, which is going to allow conjugation to occur, which is where we have 2 bacterium attached to each other and exchange nuclear information or exchange plasmids, like we just spoken about or had spoken about. So there are pili associated with some bacteria as well. And then lastly, we have a flagellum, which is singular. We can also see flagella, which is plural. So sometimes we see a single flagellum, which allows the bacterium to move. Sometimes we see many, many flagella, so that the bacteria can move and tumble and roll and things like that we'll talk more about as well. So you do need to be familiar with all of these different structures, be able to identify them on the image, and also write descriptions about them and discuss them. For now, that's kind of our general overview, and now we're going to move into some of these things in more detail, but first we're going to start out with our cell morphologies and arrangements. So cell morphologies and arrangements. And we've already discussed this a little bit, so this should be review already to you. But then now we're going to talk about it in a little bit more detail. So what we're talking about here are prokaryotic cells, because in this chapter, again, we're talking about prokaryotic cells, things that are unique to prokaryotes. So when we talk about morphology, morphology is the shape of a cell. And that's why we've already discussed this briefly. So shape of a cell is the cell morphology. And then, of course, arrangements are how they are arranged. How they are arranged in relationship to other cells. So we have the shape of the cells and then how they are arranged. So some of the shapes we've already spoken about. So we have coccus. So coccus or cocci, which is singular or plural. And again, remember that that is round shape. So nice round, spherical. Looks round to us, but spherical. We have bacillus. Recall what bacillus was, or bacilli. All right, so that's rod shaped. So remember that looked something like this. So our rod shaped bacterium. Then putting those two together, coccobacillus, or plural, coccobacilli. So what was this again? Good. So it's a combination of the round and the rod, right? So coccobacillus. So not as long as our bacillus, not as round as our coccus, something in between there. Good. So then we also have our vibrio. Do you remember what vibrio is? Our curved rod. Oh, I just noticed that we missed a piece up here. Let me erase my coccobacillus here. And we're going to write, this is a short rod, which looks something like this. And then our vibrio is our curved rod. So something like that, also called comma-shaped. Good. Then we have our kind of spiral types. So spirillum. Do you remember what spirillum looked like? Okay, so this one was the spiral. Or you could call it the wavy one. And then we have our spirochete. So this is our helical spiral. So when I think spiral up here for spirillum, I think wavy. And then our spirochete is what I think of spiral or helical spiral. So this is where we actually have the spiral going like a telephone cord spiral, what we would think of, or you can recall of a DNA molecule, for example, a helix. Good. All right, so remember that these are our cell shapes. So we started with our cell shapes or morphologies. Good. So now we're going to talk about the arrangements. And we have also spoken about this briefly as well. So our arrangements. So the first one we have is a single cell. So if we talk about a single cell, then if we use the coccus, for example, we could just say coccus. Then we would just have one singular spherical cell. If we had two, that would be diplococcus. So if we have two, and we kind of already discussed this also, they end up looking something like this. So that's diplococcus, di meaning two. Then we have tetrad. So our tetra is 4. So we end up having them in clumps of 4. which is a new one to us. We did talk about just a single coccus. We already spoke about diplococcus. So tetrad, tetra meaning four, so as four in a square. Then we already spoke about also, we have streptococcus and staphylococcus. Do you remember what streptococcus looks like? So streptococcus is a chain of cocci. So remember, we have our chain, so it would be something like this we may see. And then we have the other one, which is staphylococcus. What did that look like? A cluster of cocci. So kind of typically described as grape clusters, which is different than our tetrad. So note that the tetrad is in a square shape, and they come in those fours. And when we look at our clusters of cocci, then that would be different. We would have more than four. They're kind of clustered around. They're not in a square shape. Then we have our bacillus, which is just our singular bacillus, which is the rod-shaped. And then we can have our streptobacillus. And so streptobacillus still has the strep part, and so the strep part is chain. So this is chains of bacilli. So then it would look something like this. So chains of bacilli. So strepto or strept means chains, and staphylo means clusters. So you can keep that in mind whenever we're looking at different types of arrangements of cells. So we have the single, the two, the four, then we have chains and clusters for coccus. So all of these are related to our coccus shape. And then we have the bacillus single, and then we have streptobacillus. So the chains of bacilli. All right, so now let us talk about this kind of morphology. As we're talking about the morphology, what makes these shapes of the cell. And what makes the shapes of the cell is the cell wall. So we're going to talk a bit about the cell wall and kind of refresh our memory on osmosis and tonicity. So our cell wall is going to maintain the morphology, as we said, so maintain morphology, along with other, not without, but with other cytoskeletal elements. Meaning, if you recall, that there is a cytoskeleton, cyto meaning cell and skeleton meaning structure and shape. So within our cells, we'll have a cytoskeleton, which is going to provide structure and strength to the inside of the cell and maintain its shape. However, our cell wall does a really good job of that in these organisms. So recall that most prokaryotes have a cell wall. Some eukaryotes have it. And we'll talk about some differences in eukaryotic cells when we get to chapter 5. So recall that the cell wall envelops the cell membrane. So when we looked at the structure of the cell, we saw that the cell wall was the outermost or most superficial layer of the cell, and then just within that we had our cell membrane. Or not most superficial, because there's a capsule, but compared to our cell membrane, the cell wall is more superficial. Not all cells, not all bacteria or archaea have capsules. So that was an illustration for a cell to just show all of the different things, just like not all cells have flagella, not all cells have pili, not all cells have fimbria, but that was kind of a generalized cell. So the outermost portion, or the outermost layer of a prokaryotic cell, is going to be the cell wall. Now, however, some of them will have more superficial structures, for example, like a capsule. So it envelops the cell membrane. And then it protects the cell from changing osmotic pressure. So recall that we find prokaryotic cells pretty much everywhere. So we find them in and on our bodies. In fact, we have more prokaryotic cells in our body than we have our own eukaryotic cells. So we would find them in different areas that have different pressures, different osmotic pressures. So the surface of the skin is going to be much drier and have a different osmotic pressure than, say, inside of our intestines or inside of our stomach, which would have very different osmotic pressure. So their cell wall is going to help to protect the cell, prevent it from popping or from crenating so much, or plasmolysing so much that it actually kills the cell. So we have this changing osmotic pressure depending on where the bacteria finds itself, and the cell wall is going to help it to not die as a result of that changing in osmotic pressure. So this example also applies to the transfer of bacteria to another person. So if you think, for example, if somebody's sick and they sneeze, the bacteria had been living, say, in their nasal cavity where it's very, very moist, a certain osmotic pressure. And then as it sneezed out, say it lands on a door handle or a table. On a door handle or a table, we're not going to see the same osmotic pressure. So it's going to have to be able to withstand the changes. So let's take a moment to define osmosis and osmotic pressure. So recall that osmosis is the diffusion of a solvent across a semipermeable membrane, and it goes toward the side of higher concentration of solute. And it does this until it's equal on both sides. So diffusion of a solvent across a semipermeable membrane toward the side with higher concentration, toward the side with higher concentration of a solute. Or let's not say of a solute, let's just say of solute, or of solutes. Because it doesn't have to be a single solute. So the diffusion of a solvent across a semipermeable membrane toward the side with higher concentration of solutes. And then also we want to say that it continues until both sides of the membrane have the same concentration. So when we think of osmosis, oftentimes what we're thinking of is water, right? So we're thinking of water is our solvent, and oftentimes that is exactly what we're talking about, is the water as our solvent. So water moving through the semipermeable membrane, so the plasma membrane of a cell, going back and forth, in and out between the cell and the exterior portion, or the exterior of the cell. And this is related to the concentration of solutes. So remember, when we have an area that has a lot of solutes, we want to move water toward that area because we want to make the concentration the same on both sides of the plasma membrane. Let's define osmotic pressure as well, since we've been talking about osmotic pressure. And then we're going to talk more about tonicity. So osmotic pressure is the amount of pressure that's required to stop the flow of solvent across the membrane. So the amount of pressure required to stop the flow of solvent across the membrane. Right, so we're talking about pressure, right? The force that's required to stop this movement of solute, or solvent rather, across the membrane. So stopping this water from going across. So that pressure, and if you think about something that's really, really high pressure, you can think of it kind of pushing on it. So the force that's required to stop it from moving across the membrane. Good. So let's talk about what this exactly means, right? When we're talking about bacteria, and this should be familiar to you, but we're going to go through it again. If we think about the actual cell, and then if we think about the external environment. So what we're going to talk about is tonicity. So what is tonicity? First, what we're talking about with tonicity is the degree to which a cell can withstand changes in osmotic pressure. So the degree to which a cell can withstand changes in osmotic pressure. Good. So our cell walls are going to help with tonicity, right? So they're going to help the cells have an increased tonicity. So withstanding the changes in this osmotic pressure. So let's take a look at a cell in its external environment. So let's say we put a cell in a beaker, for example. So we have our beaker filled with our solvent. Let's say it's filled with water. And then we put our cell into the water. Let's say that this is a cell that has no cell wall, right? So what we're talking about here perhaps is a eukaryotic cell. So it does not have a cell wall. So what we have inside of the cell, right? We have lots and lots of different things inside the cell, like we already spoke about. We've got, ribosomes, and we have other things in here. Lots of stuff going on, lots of chemical reactions. So we have things happening there. But let's say in the beaker, or let's say in its environment, it's put into a solution that doesn't have that many solutes in it. So our red dot equals our solutes. So our red dot is the solutes, and then we have our cell. And we're going to say that this is a cell that does not have a cell wall. What direction are we going to see osmosis occur? Where are we going to see our net water movement? So our net water movement, so the direction that we're going to see water moving mostly is into the cell. Right, so we're going to see water move into the cell. We'll do that in blue. And the reason we're going to see it move into the cell is because it's trying to dilute all of those solutes that are inside of the cell. So remember, we want to have the same concentration of solutes outside of the cell as inside of the cell. So if the solutes are unable to move across the plasma membrane, then the water is going to be moving across the plasma membrane. And in fact, the water is always moving across the plasma membrane. And that's why we say net water movement. So we always have water going in the cell and water leaving the cell. We have this exchange all of the time. However, we're going to see the water moving more often in one direction in this situation than in the other direction. And that's why we say net water movement is going to be in toward the cell, so we can dilute those solutes and then make it equal on both sides. Now, when this happens, this is called, in this situation, the external environment is a hypotonic environment. So when we are talking about tonicity, like using the term hypotonic, you have to specify what you're talking about. What is hypotonic? Is the cell hypotonic to the water? Is the water hypotonic to the cell? So make sure that when you are discussing tonicity, you are describing exactly where that term is located. So in this case, we're going to say that it's a hypotonic environment. So right now, this is a hypotonic environment. And so what we see, hypo means less than. So our environment has less solutes than the cell itself. So if it's hypotonic, then we're going to see our net movement of water, or the solvent, diffusing into the cell. So now if we have no cell wall, what we end up seeing is, in this situation, see, I put lots and lots of solutes inside the cell, not very many solutes outside the cell. We see lots of water rushing into the cell. And in this situation, water would keep rushing in and keep rushing and keep rushing in until the cell bursts. And so because there is no cell wall, this cell will actually create holes, or have holes in it, because it's just going to burst. It's going to swell up so big that then it'll end up bursting, and then it all starts to come out because it can't withstand the pressure of the water that's being pushed into it. So it's going to burst with all of that water moving out, and then the cell will die because it's no longer living, it no longer has an intact plasma membrane, all the pieces of it float out. Now, if, on the other hand, this cell had a cell wall, so we can say that it's a bacteria, you know what, actually we'll make it a kind of plant cell. A plant cell or a bacteria, if it has a cell wall, and then within that cell wall it has its plasma membrane, then what we see, so again this is in a beaker, and inside of our beaker we have a certain solution, and inside of the cell we have lots and lots and lots of solutes, not so many solutes on the outside. Again, we're in a hypotonic environment or hypotonic medium. And then, of course, we see the water moving in. Our net water movement is in the cell, diffusing into the cell. What we see in this case is that then what we see is the plasma membrane is going to push up against the cell wall. It's going to start making the cell wall kind of push out, and the plasma membrane is going to push up against that cell wall. But since the cell wall is there, providing that cell with structure, it doesn't end up bursting the cell. So it prevents that swelling going too far, and then it doesn't burst. So it'll keep trying to push water into the cell depending on the concentrations, but that cell wall is there to prevent it from actually exploding and dying. Now if we go back up here with our no cell wall, and have our cell in here, and we take a look at our solute situation. If we take a look at this situation, which direction do you feel like our net water movement is going? So in this case, what we see is we do not have net water movement. So you can kind of tell that the amount of solutes inside of the cell are about equal to the amount of solutes outside of the cell. So recall what this one is called. This is isotonic environment. So in this case, iso meaning the same. So the same tonicity inside and out of the cell. So we have about the same amount of solutes, the same amount of pressure. So again, what we have is water movement across the plasma membrane, but it's going in and out at fairly equal rates. So there's not really a net movement in or out of the cell. If we then take a look at that situation down here with our cell wall cell, or cell that has a cell wall, and we have our plasma membrane inside of here, and we think about the solutes being about the same, we see the same thing. We see water moving in, water moving out, and that plasma membrane basically stays in the same spot. Now our third situation, if you can imagine what our third situation looks like, get a little sloppy with my beakers. We have our cell that does not have a cell wall, and in this case we have few solutes in the cell, and we have lots and lots of solutes outside of the cell. So which direction would we see our net water movement in this case? Get out of the cell. Right? So again, we want to make the concentration equal on both sides. If we're trying to make the concentration equal on both sides, our water is going to move out to try to dilute the contents on the outside of the cell, the medium that it's in or the environment that it's in. And what is this one called then? What kind of tonicity is this? Good, so hypertonic. Hyper meaning greater than. So hypertonic means that the solutes, there are more solutes than in the other location. So in this case, we have a hypertonic environment. That means we have more solutes in the environment than we have inside of the cell. If we then look at the situation in a cell that has a cell wall, as we've been using kind of the plant cell just to differentiate here. So we have our plant cell, which is oftentimes kind of rectangular. And then remember that we have our plasma membrane on the inside of that. If we have a whole bunch of solutes on the outside compared to the number of solutes that we have on the inside, then what we see is called plasmolysis. So plasmolysis, so lysis is meaning cell death, but in this case what it means is shrinks, and then plasma. So the plasma membrane is going to shrink. So then what we end up seeing is it kind of still is attached to the cell wall, but it actually is going to be away from the cell wall and all of the other areas. So it kind of shrinks, and then this is going to be empty space in here. This is kind of our empty space. We still have the structure of our cell wall, but our plasma membrane has now shrunk down and detaches from the cell wall in many areas. So it does still maintain the cell a bit because it's allowed to, it still kind of attaches somewhat to the cell wall to help protect it from these different situations, from a hypertonic environment. However, it does pull away from it because, again, we have our water movement out of the cell. So this is called plasmolysis. Up here in our cell that has no cell wall, we also see the plasma membrane change, and it kind of becomes spiky, similar to plasmolysis, but in this case, since we don't have a cell wall, it's not attached to anything. And so in this case, we call this crenation, or it's crenated. So in this case, it ends up looking really, really spiky because our water has moved out, and then our plasma membrane kind of puckers in and makes it look spiky, like a little tiny spiky ball. Good, so hopefully you recall these things from previous classes. If not, then this should be a good overview here. So recall that we have different situations occur when we have no cell wall, and compared to when we do have a cell wall, which is down here. And I didn't want to draw it this way because I thought it would be kind of confusing, but recall that when we're talking about microorganisms, a lot of time what we're talking about are bacteria. So if we pretend that this is a bacteria that has a cell wall, then we're going to be seeing this same situation but with the bacteria where the water is going to be moving in, for example, or moving both ways, no net movement in the isotonic environment, and then moving out in a hypertonic environment. So we'll see this. We, of course, see this in both eukaryotic cells as well as prokaryotic cells, but you need to keep in mind that we have a cell wall in the prokaryotic cells, most of them, and it helps to deal with these changes in tonicity, or these changes in osmotic pressure. So I am going to wrap up the video here, even though it's not the end of the section, and I'm going to do additional videos or an additional video to do the rest of the section so that it's kind of split up into smaller pieces. So just to kind of go over what we discussed here was we started up here talking about our unique characteristics of prokaryotic cells, talking about what all cells contain, which are these three things here, the cytoplasm, chromosomes, and ribosomes. We did take a little bit of a look here at our prokaryotic cells versus our eukaryotic cells, looked at some of the differences and talked about those. Also, we looked at the general structure of a prokaryotic cell here and talked about each of the different things that we are going to be going into more detail about as we move through this chapter and other chapters. We spoke about our cell morphologies. Remember, that's the shape of a cell, and the arrangements, how they're arranged in relationship to each other. And then we talked about, or we spoke about the cell wall. So we mentioned the cell wall. We're going to talk about the cell wall in more detail in the future as well, as far as the actual structure and the chemicals or the molecules and things that are a part of the cell wall. But just a reminder about osmosis and osmotic pressure and tonicity. And you should be familiar with all of these things. So if you were asked if this particular type of cell is placed in this type of environment, you would be able to describe the net water movement and what we would see, if we would see crenation or plasmolysis, or if we would see cell bursting, or what the situation would be.